Okay, so let's start. So as this is our introductory session to our paper class, I'm going to go through some of my sort of the structure for the paper class, right? So you know this paper class is a sort of it's sort of essential that you have the paper practice. That's obvious, no? Because in the A-level exam, there's a particular art to answering the questions, right? So even though you have memorized the resource book completely, particularly when it comes to answering essays and sometimes structured essays, it doesn't apply that much to MCQs. There's a certain pattern of answering that you have to master before going to the exam, right? So that's what we are going to do. But not just question answering in this paper class. We are going to do question based revision as well, right? For example, today I plan to cover using MCQs. I plan to cover 4 topics, right? Plan to cover, I mean, revise very fast the theory behind it of 4 topics, right? And plus the 2 essays, so that is going to add up to about 6 topics, right? So I'm going to teach you the theory behind it using questions, because you know, as bio is a theoretical subject, right? That is one, right? So from questions we are going to go for theory master, right? Otherwise it's going to be boring, right? We didn't start, right? So from questions, I'm going to help you to cover the theory, because as you know, this is not a theory class, right? And there's a separate revision as well. But you are in a sort of a rush, you are running out of there's a sort of a time pressure for you. So it's important that we strategically cover this. So that's one thing, right? And the second thing is the art of answering, that is, I mean, that is going to come to you automatically with doing questions. I mean, for a subject like physics, right? Questions column, we are going to master it, right? So I'm going to sort of help you do that as well. That is going to be mastered. And also, sort of, I'm going to prepare you, particularly for the A-level paper, by sort of practicing patterns, right? These patterns can come in the form of MCQ patterns, and also mostly what I'm going to focus is essay patterns. Because why? These patterns are important in the sense for the essay paper, right? For the essay paper, particularly what I did for the 2025 people, we did a 45 essay plan in the final 2 months, 45 essay plan. So from this 45, there was at least 4 in the A-level paper itself. Paper එකට ආව 4. So if you practiced, I mean, my 2025 people went through this plan, and you know, 6න් 4යි answer කරන්න තියෙන්නේ, right, in the paper. So they got almost similar questions in the essay paper, right? So patterns practice kalla, you can go to the paper in a stress free manner, right? The amount of stress, particularly before the essay paper, is low for you, right? It is because of you are studying it in a systematic way. There's no time for you, right? And also, it's going to reduce your stress, right? So these are the things that are going to, I mean, I'm going to give you as a part of this sort of paper class package, right? And also with your papers are marked, right? Marked and given, so you can check, do a self evaluation, what went wrong, right? Particularly in the essay paper, when you receive your marked essays, compare it with the marking scheme I give, right? Mandina marking scheme එකයි what you wrote, please compare and see. Sometimes you you might think the marking is a bit too tough. Paper එක mark කරලා ලකුණු දීලම නැහැ, right? You might think the paper was marked a bit too tough, but that is for your own benefit in the long run, right? That's for your own benefit in the long run, because we need to prepare you, නිකන් නලවල හරියන්නේ, right? It will appear like We are not giving you marks, but with that sort of tough practice, you are going to get marks in the paper, right? So those are the few things that I want to sort of mention as the as on the first day of our paper class, right? And if you are struggling with sort of the deeper theory parts that is not covered by that is not covered in this paper class, the solution for you is to join the revision, right? To join the revision. And few things about our revision. We are currently doing genetics, right? Genetics day 3 we are about to start. 2 days of genetics we have done, right? So what is a bit special about our revision class is, as you know, most revision classes start from the first lesson, no? Right? First lesson, it's a revision. So first lesson onwards, they go from introduction to biology and chemical base. But what's sort of, let's say what many students find so boring about, not boring, but a bit, let's say, unprotective about these revision classes is, as you know, first, second, third lessons you have done so many times, right? Because they are done in the first year, ne? Right? First year of your bio life. So in the first year, your theory classes are teaching very well, and they are going very slow. They are doing so many extra questions and everything. So these few first lessons, you have sort of, I know whatever level you're in, you have mastered. Ne? Then ඇති වෙලා තියෙන cellular and chemical basis, right? Evolution and sometimes plant form, right? And you have faced some term tests. Term test with face color, at least 2, 3, right? So we are going to do the revision of these lessons, but there are so many other lessons that you might have skipped, or the theory class went a bit too fast. Like, push, gala, they have done genetics within 2 days because the class was running out of time, right? So, lessons like, so after genetics, we are going to start molecular biology, right? We are going to start molecular. Then we are going to do animal form, right? So, our revision class, what I'm going to focus is the high yield, right? And also the untouched topics, right? Question heavy and untouched topics. I'm going to focus on first because you are not yet stressed. තාම එච්චර stress එකක් නැහැ. When you come to the last 3 months, මේවා කරන්න ගියොත් again you are going to go in the same hole. ආපහු හරියට කරගන්න බැරි වෙන විදියට stress වෙලා these sort of let's say tricky lessons are going to be a problem for you again. ආපහු then revision class ඇවිල්ලා ආපහු those lessons are going to be a problem, a headache, stress. Okay? So that's what we are going to do. Early in the revision class, we are going to cover these critical topics, right? And then we can come back to chemical basis, evolution, plant form, things like that. Okay, clear about that? Okay. Any questions, online people? Everyone has got the paper, right? Can you hear and see me clearly? We are on a new location. It's going to change from next week, right? So there can be a bit of a darkness issue, but from next week, those problems will be solved, right? Okay, so let's move on to the paper discussion itself, right? We are running out of time. Okay. So the paper discussion, I'm going to do it in 3 parts, right? First, let's start with the structured essay. Yeah? First, let's start with the structured essay. Let's start with the structured essay. All of you can hear me and see me, right? All good. All right. Let's start with the structured essay. It's a bit longer than a typical structured essay, right? ටිකක් දිගයි, but I gave only one structured essay because I am going to take a bit of time to discuss this. Okay. So without further ado, let's quickly get into it. Online people, questions? No, but all good, right? So first question: what is the cause of threat to natural resources? So these are introduction to biology, right? So what are the threats to natural resources? You can send me, right? I want some interaction. Yes. What are the causes of risk to biodiversity? Sorry, natural resource. Sorry, natural resources. Tell me what is the definition of natural resources? Resources of material. Okay. So SCQ, right? This here you can see, right? So we are starting with the SCQ, right? Question number one. What is the definition of natural resources? Right? What is the definition of natural resources? Sources of materials and energy, right? Right. These are important topics, right? So natural resources can be material or energy. Yeah? I'm going to take a bit of time. Material or energy. It could be two things, right? Natural resources can be materials or energy forms, right? Found naturally. Obviously, natural resources are found naturally, used in everyday life and for economic development. So they are used as a person, right? As a person of the society, you are using natural resources, and for as a country or a factory, maybe you are using them for economic development. So, what are the cause of threat? There are few threats, right? That's given. Increasing growth rate of human population, overuse of natural resources, right? So it's the same thing, right? So how do we write that? What is the threat? The threat is increasing growth rate, right? I have given you two rules, huh? You need a bit of a descriptive answer, ne? Two rules. Increasing growth rate of human population, that is going to lead to what? Overuse. Simple, ne? Right? So this is the answer. What is the main threat? Increasing growth rate of human population leading to overuse of natural resources. Right? Coming to second question, take the two consequent processes, increase in human population. You are missing half of the answer. You are missing half of the answer. Let's say growth rate can be a keyword, right? Growth rate can be a keyword depending on the marking scheme. Depending on the marking scheme, they can give you growth rate as a mark, right? So growth rate කියන එක නැත්තම් sometimes you can lose. And overuse is a keyword. Overuse අනිවාර්යෙන්ම ඕන. Right? It's not just the use. Uh, you have to overuse it. නේද? You have to overuse it. Second one, part A, state the two consequent processes that happen during the lifespan of organisms. So we come to consequent processes that happen during the lifespan. Yes, what are the answers? What do you think? 2A. එකකට පස්සේ එකක් නේ. Consequent කියන එක one after the other. What are the consequent processes? Yes. Yeah, very good. Right. Growth. After growth, what's going to happen? There's going to be a development. There's going to be growth. Then there's going to be development. What is the definition of growth? Quickly, right? Highlight these things if you haven't in the resource. Huh? I want you to have the resource book turned all the time. Right? What is growth? Irreversible increase in dry mass. Growth is dry mass increase. What is development? Irreversible changes that occur during the lifespan. Right? Irreversible changes. Okay? So that is development. These are consequent. Growth happens, organism develops. Growth happens, organism develops. All right. Question number 2, part B. How can living organisms be distinguished from non-living entities? Yes. How can you differentiate living from non-living? ඒකනේ basically අහන්නේ. How can you differentiate living from non-living? Yes, there's three rules given. Three rules given. So what are they? They are asking a detailed answer. So they are asking a detailed answer. Any suggestions? Suggestions for answers? මොනවද ලියන්න පුළුවන් අපිට? How can living organisms be distinguished from non-living entities? By the characteristics of organisms. No? By characteristics of organisms. Right? By their characteristics. So I want you to list down all the living characteristics. What are the living characteristics? Characteristics of a living organism? Order and organization. Right? I'm not going to write all. Right? What are the living characteristics? Order and organization. What else? Metabolism. What else? We just talked about growth and development. What else? Irritability and coordination. Irritability and coordination. What else? Right? Reproduction, adaptation, right? All these things all together, right? A living organism is able to express all these. Non-living entities do not express these. So those are, that is how you distinguish living from non-living. Simple, no? Any questions? Come to question number 3, part A. 3, part A. How does water function due to its high specific heat? So we are coming to water, right? I'll do a small revision about water. What are the 4 main characteristics of water that you need to know? What are the 4 main characteristics? Can you see up to here? Yeah. What are the 4 main characteristics of water that you need to know in your syllabus? There's a suggestion. Living organisms. Hmm. Living organisms. Yeah, that could be added. Yes, right. After you have listed down all these living characteristics of living organisms, you can add, right? Only living organisms display all these characters simultaneously, right? Yeah, that's a good point, right? Only living organisms express all these simultaneously. Okay, right? For example, if you take a virus, what do you know about viruses? Virus වලට මොනවද කරන්න පුළුවන්? Only single thing. They can only reproduce, right? Viruses can only reproduce. They don't have a proper order and organization. They don't have any metabolic activities outside host cells. They don't react. They don't adapt, right? The only thing is they can reproduce, photocopy, 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 right? So if you want something to if you want to categorize something as living, all these must be expressed what simultaneously. That's a good point, right? So you have to say living organisms display all characteristics simultaneously or at certain point, right? Or at a certain point. Can you see? I am strictly sticking to the resource book, okay? Because this is not a theory class. I'm strictly sticking to the resource book, right? Because why? There's no time, and also that's what's going to give you marks. That's how you answer papers. This is a paper class, right? So I'm sticking to the resource book strictly, no? Right? Because if it was a theory class, I can take some time and explain each of these things, but it's not. Okay. Okay. So characteristics of water. What are the 4 main characteristics of water that you need to know? cohesive behavior. Cohesive එකත් එක්කම we have adhesive also, right? Can you see this? And yeah, cohesive. Then moderate temperature. What is the other one? Yes, expansion upon freezing. Expansion upon freezing. Right. Finally, what? Versatility as a solvent. No, it's a very good solvent. These four නිකන් අර you have to parrot වගේ මතක තියාගන්න ඕන, right? Because these are basic things you learned, right? So what are they asking? How does water function due to its high specific heat, right? So that comes under what? Ability to moderate temperature, no? Ability to moderate temperature. It comes under this, right? So due to high specific heat capacity, what is the application? High specific heat capacity. So when we talk about ability to moderate temperature, there are few things. Okay. Moderate temperature. So I'll erase the others and talk a little bit in detail about this moderating temperature. Okay, can I erase the others? Right. So under this ability to moderate temperature, we have sort of few subtopics. What are the sort of subtopics that is coming under this? Yes. Yes. First one, we can say this sort of thermal buffer story, thermal buffer කතාවක් තියෙනවා නේද? Right? So it's one of the features coming under ability to moderate temperature. So thermal buffer. Then heat of vaporization story එකක් තියෙනවා නේද? There's a story about high heat of vaporization. So under this thermal buffer, what do you have to say? It is able to what? Absorb or release a high amount of heat. So therefore, a system that contains a lot of water, you can't change its temperature very easily because the water can absorb or release a lot of heat without changing its what? Temperature. So temperature remains constant. That's what we are saying under thermal buffer. No? So that you have to mention. What is coming under vaporization? Two things. That is sweating in sort of mammals and including human, and transpiration in plants. So I'm just reminding you, you have to organize your information like this. නැත්තම් මේ ඔක්කොම නිකන් paragraphs විතරයි in the resource, right? You have to organize, right? So what are they asking? How does water function due to its high specific heat? So we are asking high specific heat, that is coming under what? The thermal buffer part, no? High specific heat කියන එක තියෙන්නේ thermal buffer section. So what can you write? You have to say water will function as a thermal buffer in the living system and aquatic bodies during temperature fluctuation, right? Right. Due to high specific heat, water will function as a thermal buffer in living system and aquatic bodies. Got it? Right. ඒකට තමයි rules දෙකක් තියලා තියෙන්නේ. You have to be bit descriptive, right? When people, I know it's getting a bit dark, right? That will be solved next week, right? Because it's a new location that we are doing classes at. Okay. Any questions, online people? Moving on to the next question, right? Up to here, no questions, right? What is the next question? What proportion of elemental composition of living matter is calcium, phosphorus, sulfur, and potassium? Calcium, phosphorus, sulfur, and potassium. So if you come to the elemental composition, if you come to the elemental composition of living organisms, there's a big four, ne? හතර දෙනෙක් ඉන්නවා dominating, right? There's a big four, and there are other small elements. So how many in total? If you take the human, how many total elements can you find? Yes, in humans, how many in total? We are in question number B. Yeah, there's a small issue with the numbering. Yeah, 3B, no? Yeah, 3B. Okay, 3B. So if you take humans, we have 25. Plants now how many? Yes, plants now how many? 17, no? 70. Okay, if someone missed the point during the temperature fluctuation on Earth, for the previous question, no? Yeah, that is essential, no? Due to high specific heat that is in the question. Thermal buffer is a keyword. You have to say thermal buffer. In living system and aquatic bodies, 2 more points during temperature fluctuation. That's what it matters, no? නැත්තම් temperature, if the temperature was constant, we don't need water to act as a buffer, no? Okay. So humans and humans 25, plants 17. What are the big four? Big four elements: carbon, hydrogen, oxygen, nitrogen. If you take a living system, 96% is these four, right? 96% is these. Can you see now we are doing revision? Because why? You don't need a sort of, you don't need to go into detail into these early lessons because you know this. These are simple things. There's nothing for me to teach here, right? So 96% is are these. Then there's a another, let's say there's another four that remain, that compose the most of the remaining 4%: calcium, phosphorus, potassium, and sulfur. Calcium, phosphorus, potassium, and sulfur, most of the remaining 4%. Most of the remaining 4% by these, right? So what are they asking? What proportion of elemental composition of living matter? So what you have to say, make up most of the remaining 4% of the mass of an organism. Okay. Then additionally, I want you to sort of remember them right now: sodium, chlorine, magnesium, boron, cobalt, copper, chromium, fluoride, fluorine, iodine, and iron are found in minute amounts. So you know, included into this 4%, මේ 4% එක ඇතුළේ ඒවා තියෙනවා. But most of it is by these four, නේද? Clear? Moving on to question number 4A. 4A. Two branched, what are they asking? Two storage branch polysaccharides. What do you have to be? They have to be polysaccharides, right? No, if you have mentioned just four, that's not true. No, because මේ හතරෙන් නෙවෙයිනේ all four like entirely this 4% is not by these four. අර අනිත් ඔක්කොම ඉන්නවනේ cobalt, chromium, fluorine, iodine, blah blah ඒ ඔක්කොම ඉන්නවා. So you can't say four. You have to say most. Because අනිත් අය චුට්ටියට හිටියට they also contribute to this 4%. Right? Right. So polysaccharides, I'll do a small, let's say, small revision. How can you classify polysaccharides? You can classify them structurally or functionally, no? Based on their function or structure, you can classify. How can you classify structurally? As linear or branched, right? Then functionally, how can you classify? As storage. Sorry, we don't call this structure because it can be confusing, right? We better call this based on architecture, become architecture again, right? Architecture. Yeah, the online people. Architecture. Functionally, you can have storage and structural. Structural. Yeah, structural. Right? Structural. This is the basic classification. So, what are they asking? Storage and branched. What are the So let's think about the storage ones first based on function. Storage, what do you have? We have basically starch and I'll use a different color. All right. I'm not going to list out all the polysaccharides. Okay. So storage, what do we have? Starch, we have glycogen. No? All right. Then what are they asking? Branched. So branched, what do we have? We have glycogen, which is one of the branched. And if you take starch, has starch is composed of two components. No? Starch is composed of amylose. There's a sort of a polysaccharide strain and amylopectin. No? Nail. Right? There's amylose and amylopectin. Meaning amylose is linear. Amylose is linear, amylopectin is branched. Right? So what is the proper answer? How do you compose this? Because they are asking storage, you definitely have to go with the starch and glycogen. But don't be confused because there's a linear component in starch as well. Right? But as a whole, we can take starch into here because it's storage and there's a branch component. So starch and storage are the starch and glycogen are the answers. Well, starch and glycogen. Okay. Oh yeah. All the mm, so linear and branch storage. Yeah, you can put inulin under storage, but we don't know. You don't have the structure of storage. Inulin, the inulin. We don't learn about the structure of inulin. No? So because they are asking bit of architecture also, only name the things you know the architects. Structural architecture you know. No? So Bayanatu you can write. Right? What else? Linear. Yeah. Inulin is linear, yes. But it's not in your resource. Yeah. Okay. Moving on. B. Yeah. For B, what is the question? State a physical difference between saturated fatty acids and unsaturated fatty acids. Right. I'll come here. All right. Saturated fatty acids and unsaturated fatty acids. If you take a saturated fatty acids, all the carbons in the hydrocarbon chains of the fatty acids are having what? Single bonds. Saturated, no? Saturated by bonds. These are basic things you know, right? Carbons in a unsaturated, there can be double bonds. There can be double bonds in a unsaturated. Okay. No, hold on. So they are asking a physical difference. Read the question very carefully. They are asking a physical difference. So how do you, what is the physical difference that happens when a fat is containing saturated fatty acids, or if the fat is containing unsaturated fats? Physical property. What is the physical property? Yes, saturated fats, because they have saturated fatty acids, they are solid at room temperature. Very, very important to say, at room temperature, you have to say at room temperature saturated fats are solids at room temperature, and the opposite for unsaturated, they are liquids at room temperature. Animal fats such as butter comes under saturated. So you might think, why is butter melting? Yeah, ලංකාවේ නම් butter meltනවා නේ. In the, if you keep butter outside the fridge, it's going to melt. Why is that? Why is butter melting in room temperature in Sri Lanka? Because our room temperature is about 31, 31, 32 again. What is the sort of the accepted value for room temperature? 25, 25. So 25 already they won't melt in a, so we take 25 as the standard room temperature. But our room temperature in Sri Lanka is about 31, 32. On a bad day, 33. Very hot day, right? Can you see up to here? Online board you can see. Yeah. So physical difference, just one rule නේ දීලා තියෙන්නේ. එක line එකයිනේ දීලා තියෙන්නේ. So that's they are expecting a short answer නේද? Right? ටක් ගාලා evaluate, quickly evaluate what you have to write. Otherwise you are wasting time by thinking and writing. දෙකටම you are going to waste time. So take a look at the question and sort of let's say evaluate what you have to write. වැඩිපුර ලියන්න දඟලන්න ඕනේද නැත්නම් are they expecting a short answer? Moving on, name an amino acid that has an asymmetric carbon and draw and label it. Okay. What are the parts of an amino acid? What are the parts of an amino acid? Question number C, right? Part C. What are the parts of an amino acid? Yes, there's a central carbon. There's an amino group, NH2. There's a, let's say, always there's a hydrogen group. There's a COOH group. And there's a variable R, right? So label this. But what are they asking? The symmetrical one. No? To be symmetrical, what you need to have? You can fold it and it has to be sort of mirror image. No, mirror images. You have to be able to superimpose kind of. No? So what is the only situation that you have a symmetry? In the case of what? What is the amino acid? Yes, what is the amino acid that is symmetrical? What is the amino acid that is symmetrical? Hmm? Glycine, glycine. These are basic things. Glycine is the amino acid that has a hydrogen in the place of the R group. Has a hydrogen right? So glycine, I'm not going to label, right? Central carbon, hydrogen group, amino group, carboxyl group, hydrogen atom in the place of the variable R group. Memai, how you draw an amino acid? What is this amino acid? It's glycine. It's glycine. Next question. Why did scientists consider using other forms of radiation instead of light rays? Right. So this sort of question won't be asked in the A-level paper, right? I sort of put it just for revision sake. Like a part of a, right? If you like, if you give this paper to someone from another country, maybe even a biologist කෙනෙක්ට දුන්නත් they might be confused. What මොකක් ගැනද මේ අහන්නේ? Radiation for what? Right? Instead of light rays for what? To do what? To take an X-ray or whatever. No, right? Because you know, it is, we are talking about what? Microscopes. We're talking about microscopes, right? Because why? Light microscopes have a limitation. No? Question number 5A. Light microscope have a limitation. No? What is that? That limitation is resolution power. Resolution means what? It's the clarity of an image, right? How sharp, and again, when you want the TV image to be sharper, appear remote sharpness, right? So that sort of sharpness or the fine borders come with what? The resolution power of the wave that we are seeing, right? So if you want to make an image sharper, we have to use a sort of radiation with smaller wavelength. This is lambda. What is a symbol? I don't know. In chemistry and physics, you use this symbol for wavelength. No, this is wavelength. When wavelength increases, the resolution will decrease. So what do you have to do? You have to decrease the wavelength. You have to decrease the wavelength. So that's what scientists did when they switched from light rays to electrons. No? So from light rays to electrons. We are talking about electron microscopes here. That's why scientists invented electron microscopes. So where do you have that answer? In the resource under microscopes, so there's two rules given. Question again, there's two rules. Yes. The limitation imposed upon the resolution power of the light microscope is by the wavelength of light. You have to say that. There's a limitation on the resolution power of the light microscope by the wavelength of light. So the wavelength is too big. There's too much sort of distance between the two crests. I'm going to teach you a little bit of physics. There are sort of situations, positions in the wave. No? Between two similar situations, you say it's a wavelength. No? Right? At least two lower points, two higher points. That you have wavelength. I think I'm right. All right. This is a wave. So that's why scientists switch to electrons from light. So you can say this. Then limitation imposed upon the resolution power of the light microscope is by the wavelength. Then resolution power is inversely proportional to the wavelength. Okay, clear. Moving on to the next question. What are glyoxysomes? State a function of them. B. 5 B. Question number sub question 5B. Glyoxysomes. Come to organelles. Page number 32 resource book. Page number 32 resource. right? මේගාම resource book එක highlight කරලා නැත්නම් if you haven't highlighted, if you haven't gone through, right? This is the time now. This is the time to customize the resource book. There's no other time, right? If you haven't, or if you have just bought the resource book, right? I really don't like when sort of, I don't think that happens in sort of classes now, when sort of these teachers give the resource book printed as their own, right? As part of their tune. You're not allowed to do that. Yeah, you're not allowed to print the resource book and sort of publish it with your own name, right? It's illegal actually. So I don't give such tunes, right? In my tunes, there's only past paper questions and diagrams, right? I don't give the resource book because you buy a resource book, you nicely make it yourself. I mean, your own, right? Customize it. It's your own private property, your resource book, right? For this 2.5 years, it's your private property. So glyoxysomes comes under what? Peroxisomes. No? Glyoxysomes comes under peroxisomes. What are peroxisomes? Some few points about peroxisomes. They contain oxidizing enzymes. Oxidizing enzymes. So what do these oxidizing enzymes do? Peroxides. They break down peroxides into more neutral forms. They break down peroxides into more neutral forms. Peroxides are toxic. Peroxides are toxic. They can damage chloroplasts. They can, I mean, right? They can interact with other enzymes. They are always very damaging. So peroxisomes are there to remove these, right? Also, in animal cells, also these are very sort of toxic. We call them reactive oxygen species, right? මේ ඔයගලන්ට නෑ particularly for atherosclerosis අපේ අර cholesterol deposit වෙන්න ඉන්න arteries, for that also there's a connection with these peroxides, reactive oxygen containing species, right? Whatever. So the job of a peroxisome is to break down these peroxides. There's a specialized peroxisome found in only in plants. Very, very important MCQ point, right? Only in plants that you can find glyoxysomes, right? Only in plants and in the fat storing tissues of plants. So what do? They break down fatty acids into sugar. These are the MCQ points that you need to know, right? Or the ඔය දැනගත්තා you know glyoxysomes found only in plants, found in fat storing tissues. They convert fatty acids to sugars. Simple. So state of function of them. What are glyoxysomes? So the question, what you have to write? Specialized peroxisomes called glyoxysomes are found in fat storing tissues of plants. They convert fatty acids to sugar. ඔය ටික කිව්වා නම් you can fill out those 3 rules. Full marks. Full marks. Clear. Any any doubts? Shall we continue? Right. Part like big section, capital B section, right? So we are done with part A, right? Don't mind those numberings, huh? Right. Don't mind those numberings, right? Okay, so we come to a little bit of cell cycle, right? Don't worry, we are going to do cell cycle in detail in the revision class, and right? So, but anyway, I don't think you need to stress that much. Nee, cell cycle again, you don't need to stress that much. You have learned about it so many times, and by now you need to know there are bigger topics. මීට වඩා cell cycle එකට වඩා අමාරු දේවල් there are in the syllabus. Female reproductive system, right? What else? More difficult things. Process of urine formation, um, process of protein synthesis, molecular biology, right? There are much more difficult topics, right, rather than cell division. So what is the question? Events that takes place in the S phase. Okay, it's raining here, so let me know if there's any connection issues. It's raining here, so let me know if there's any connection issue. Okay, if you have any connection issue, I will make the recording available to you. Okay, I will make the recording available to you. Check cell cycle එකට ටක් ගාලා switch යමු. Let's do a small quick revision about the cell cycle. What do you need to know about the cell cycle? If you take mitosis, right, mitosis, and I mean any sort of cell cycle, what is the definition? From the end of one cell division to the, what is the definition of a cell cycle? Events that take place in the life of a cell from end of one cell division to the end of the next cell division. So the entire life cycle of a cell is the cell cycle, no? Only a small part of it is actually the division. That idea you have. Cell cycle එක කියලා කියන්නේ only a very I mean sorry, cell division කියලා කියන්නේ it's a very small part of the cell cycle itself, the life cycle of a cell, right? So what are the parts of a cell cycle? Two main parts: the interphase and the M phase. So what are they asking? S phase ගැන නේද අහන්නේ? So interface is 90%, no? Right? This is the interface means the cell is just living. The cell is just living. It's like us. We are eating, we are sleeping, we are getting bigger, we are growing. So we are just living. Only a small part of the life cycle of a cell or the cell cycle is the M phase, right? So that's why we say interface is 90%. So what are the parts of the interface? Right? What are the parts of the interface? We have a gap phases G1, you have S, and you have G2. Right? Let's focus on this S phase. S is short for synthetic. What are the main things that is happening in the S phase? What are the main things that is happening? There are three things happening in the S phase. Three things happening in the S phase. Right? What is that? DNA replication. Right? Maybe they organize the information, nah? Right? Organize the information. නැත්තම් it's going to be very difficult to memorize, and also you are going to miss points, right? Now, can you see in that single sentence in the resource book under the synthetic phase, there's a lot of information that you have to remember. DNA replication occurs. Then histone synthesis. Histone synthesis happens. What is the final thing? DNA and the histone wind around to form chromatin. All these happen. Excuse me, 1 minute. all these three happen in the S phase. Okay. So they are only asking two events, but we are not going to limit ourselves with only two, right? All three, please remember, right? All good, right? All good. Any issue? No? Any issue? No? All right. Next question. We only did one question, right? Only the S phase thing. Okay. Explain a chromosome present in the prophase of life cycle. All right. So it's a bit of an unconventional question. Good for revision, na? Right? Explain a chromosome in prophase. අපි කොහොමද මේක explain කරන්නේ? How do we explain a chromosome? Right? Question number B. In prophase. So prophase comes under M phase, no? Prophase comes under M phase, no? What are the මතක නැත්නම් it's a bit worrying, right? What are the sub phases of the M phase or the mitotic phase? What are the sub phases? Prophase, prometa, then what? Meta, anaphase, and telo, no? So they are asking you to describe a chromosome in the prophase. How do we describe? Yes. Any answer? Any? So there's three rules of answers that you have to mention. Right? I'll give you the answer like this. First thing you have to mention, chromatin get condensed. Chromatin gets condensed to chromosomes. First, you have to mention that. You have to mention that, right? We don't have something called chromosomes in a normal cell. Normal interface එක in a cell එකක we don't have something called chromosomes. It's like our luggage, right? You don't pack your all your clothes into a suitcase when you are at home, right? When you are just going about in your normal life, you don't pack all your clothes into a suitcase, right? So when you are at home, all of your clothes are in the form of chromatin in the cupboard or on the chair, on the bed or wherever that you can easily access, right? You can easily get it. But when you are going to travel, you pack them nicely into a suitcase. So that's like that. When the cell is about to divide, the chromatin is nicely packed into chromosomes. That's how you divide them equally. So first thing, yes, chromatin gets condensed into chromosomes. Then what do you have to say? Chromosomes appear with the two sister chromatids attached at the centromere, right? Two sister chromatids attached at the centromere. What do you mean by this? Right, because in the synthetic phase, we have copied the DNA. No? So the DNA exists as our iconic X-shaped structure, no? Right? So these are identical copies. These are sisters. Sisters are attached where? At the centromere. Right? At the centromere, the sisters are attached. So that's another thing that you have to mention about the chromosomes in the prophase. Then you have to mention the chromosomal arms. Right? These chromosomal arms are attached by a special protein called cohesin. Maker. These chromosomal arms are connected by what protein? Cohesin. Online people, can you see my green color? Cohesin protein. So what do you have to say? The full answer is then chromosomal arms by cohesin protein. Okay. Chromosomal arms are attached by the cohesin protein. So what are the things you have to mention? Chromatin are condensed by shortening and thickening into chromosomes. Then chromosomes appear with two sister chromatids attached at the centromere. Chromosomal arms of the sister chromatids are attached by a special protein called cohesin. ඔය ටික කියන්න ඕනේ. Those you have to mention. Yes. Yeah, that is accurate. In the chat, any question you have, you can ask. Huh? It's raining here. Any lags or drop in quality? Right. Next question. State the differences in cytokinesis between animal cell and a plant cell. Now, so at the end of the M phase, we have what? At the end of the M phase, we have what we call as cytokinesis, right? So, what is the definition of the M phase? What is the definition of mitosis? Very, very important. Mitosis is only a what? Mitosis කියලා කියන්නේ it's only a nuclear division. We only talk about the division of the nucleus in all these. Right? The definition of mitosis is what? It's a nuclear division which gives rise to genetically identical daughter nuclei. Nucleus දෙකක් විතරයි we get from mitosis. Only when we have mitosis plus cytokinesis. We have two identical what? Daughter cells. නැත්තම් we are only left with daughter nuclei. Because why? Mitosis only talks about the nucleus. So we need cytokinesis to have two daughter cells.
Renai, how is the life? Who else you have? You have Nangi.
So cytokinesis is different in animals and plants.
What is
right? Cytokinesis
What
is
is
different in animals and plants. Come to page number 41.
Third.
Come to page number 41.
SLIIT or SLIIT, no?
Page number 41.
SLIIT
There are two ways of
SLIIT.
how these daughter nuclei are separated into the two daughter cells. Right?
Maradan?
I hope you have seen diagrams of this. I'll draw it for you, right?
In which year he did fellow?
In animal cells, there's something called a cleavage furrow. There's something called cleavage furrow. It's like this. Cells දෙක තියෙනවා, right? They are sort of like a crack.
That because of the මගේ මේ
that is pulled. Remember the plasma
Hasin
membrane.
අක්කගෙනෙක්ගේ පුතෙකුත් සිටියා St. Joseph, but he was a swimmer, national swimmer at that time. I don't remember his name. I think he did not.
There's a sort of what we call as a furrow. That is
you had to do London.
Right. So these are the two cells that are the plasma membrane is sort of squeezed in, sucked in by the actin filaments. So you are ended up with this is what? Cleavage furrow. In plant cells, there's no such thing because why? There's a sturdy structure around it. No? There's a sturdy structure around it.
I know.
What is that? Cell wall. There's a sturdy cell wall around it. So they can't pull their plasma membrane in like this.
Now you three continue.
So what they have to do is they have to generate a new cell wall.
This class is comfortable. Then the other one.
We have
Oh,
to
that was
make
out.
a new cell
I got
wall
it. Oh, like that.
in the
Okay.
middle of
Real.
this big cell. So
So,
that's how
Thursday
they
ki
are
class
going to
with them.
separate the cell
You had
into two,
classes on Thursday, no? That's what happened. No?
right? This is called what? So
Rest of
these
that
are the
class.
two
Which one?
individual plasma membranes, the blue color.
Which class? Which
This is called
which
what?
class?
Cell. Yes.
Class?
Cell plate.
Ah, okay. Come.
So in plants, we have a cell plate
Take
formation.
පුළුවන්
In
but it take time අර මේ record කරලා edit කරලා
animal cells, we
ඉනාවෙන
have
කපලා පුළුවන් 3 days on
cleavage furrow. This cell plates originates from vesicles from Golgi.
just මොනවාරි කන්නේ
Golgi තමයි මේකට අර materials ටික දෙන්නේ නිකන්. So we are making a wall, no? Like Trump ගේ Mexican wall එක වගේ. So who is supplying the material? Hardware බඩු දෙන්නේ කවුද? Golgi. Golgi provides the cement, not cement, right? What is the sort of the cement that you need to make this wall? Cellulose. It's a cell wall, cellulose, semicellulose. You need a bit of what? අපිට middle lamella එකකුත් තියෙනවානේ, bit of pectinose, right? So Golgi provides the
you want
vesicles for
and even chemistry have
cell plate.
no the working
So state the differences between cytokinesis between animal and plant cell.
you paid me no
Animal cells, a cleavage furrow forms. In plant cells, a cell plate forms as a result of vesicle produced by Golgi.
what do you want that is like
All clear, right? So fast. No. I'm explaining everything around, right? I'm explaining everything around the theory part, right? And I'm here. Okay. Clear about all this. We have to go to a little bit of metabolism now. Metabolism will demo, right?
Recording in progress.
So, right. Metabolism is the sum of chemical reactions taking place in living cells, right? So, what is the question asking? Types of phosphorylation. Phosphorylation කියන්නේ මොකක්ද? Addition of phosphate. Particularly, what phosphorylation, what addition of phosphate are we considered about? We are concerned about in living. ADP phosphate එකක් දාන එක නේද? ADP phosphate එකක් දාන එක. That is the phosphorylation that we want, right? So question number 2A. What is the reaction of phosphorylation? Adenosine diphosphate වලට inorganic phosphate දාලා we get adenosine triphosphate. This is energy currency, right? This is what our phosphorylation means. So what are the ways that we can do this phosphorylation? What are the ways that we can do this addition of phosphate? We can do it using light energy. Photo phosphorylation. We can do it using light energy photophosphorylation. We can use it. We can do it using high energy molecules, right? We can use it. You can do it using high energy containing complex molecules. That is called substrate phosphorylation. You can say substrate level phosphorylation or substrate phosphorylation. We are using complex molecules. We are breaking down complex to simple. In the process, energy is released. What is the third way? Oxidate. Oxidative phosphorylation by oxidation. So here it's light energy. Here it is complex to simple breakdown that is going to release. Here it is what? Release of electrons. Electrons are getting released, so that electrons energy are going to be used to generate ATP, oxidation of molecules. So what is the question? Write the type of phosphorylation seen in plants. Obviously photophosphorylation can be seen, right? What plants are the main, let's say, photosynthetic organisms, dominant. Substrate level, is there substrate level phosphorylation in plants? What is the example for substrate level phosphorylation? Respiration, cellular respiration. No? We are breaking down glucose, glycolysis level, right? We are breaking down glucose. So yes, plants also do cellular respiration. They respirate. Oxidative phosphorylation, yes, they produce. They can do oxidative phosphorylation, right, in their mitochondria. So all three can happen, right? You can write all three. Right? Write all three. Rules dekak තියෙනකෙන් don't be confused. Rules dekak තිබ්බ කියලා don't be confused. Right? What is the definition of phosphorylation? Production of ATP within cells. Making ATP. How do we make ATP? Photo substrate oxidate. Simple. Next question is a little bit about enzymes, right? So this is how you want to do it. You have to be we are jumping too fast from topic to a topic. No, right? Because what you have to familiarize, you have to be comfortable jumping here and there in the resource, right? Resource book, you have to be sort of, it has to be your sort of a, have you heard of something called mind mazes, mind castles, right? There's a memorization technique. Have you watched Sherlock, the TV series? Right, there's a special episode, right? They talk about there's a billionaire who is able to memorize so many things, right? His memorization power is very, I don't exactly remember the line. So how he memorizes things is he makes a mind like a castle, right? So in a castle, he's able to store different information. So he's visually walking around the castle and in different rooms, he has stored different information. Different subjects may room make this subject. So when he closes his eyes and goes to this room mentally, he climbs the staircase and goes to that room and he opens the door mentally, he's able to access all that information because he's visually, he has made a massive imaginative sort of a mind castle or something. It can be anything, right? Watch that episode. Have you know this Sherlock Holmes, right? Sherlock, there's a TV series, right? It's good. There's only about I think 12 episodes. Only 12 episodes, right? It's good for brain stimulation. Na? It's not a brain rot right? It's not going to sort of, it's not going to distract you. You need a lot of brain power, right, Sherlock? It's good to, you have to focus on the story, right? Watch it. Online people who has watched Sherlock. These are the ones you have to watch, right? Anyway, so what I was saying is you have to be comfortable jumping here and there in the resource. You have to have the sort of the entire resource as a map, right? You have to be able to access different topic very fast. That's how you are going to be sort of in the long game, right? In the long game, you have to be a master of the resource. Anyway, the next question, how can pH lead to a decline in enzymatic activity? Question number B. Sorry, question part B. pH, pH and enzymes. pH relates to sort of the acidity and the basicity. No? Enzyme activity is basically what? An enzyme combines with a substrate. Then you get a product. This combination is entirely dominated by what? Bonds. bonds forming between the enzyme and the substrate, right? So the environmental pH where this bonding happens is very, very influential. It can control the bond formation, right? For example, in your syllabus, we consider about two enzymes, right? We consider about a enzyme and a small enzyme. I'll draw it just to remember, right? So the pH scale is 1 to 14. You know, 14 is the most basic, 1 is the most acidic, right? So in the middle 7, pH is neutral. So if you take your stomach, right? If you this is the rate of reaction on this side, right? If you take your stomach, it has a lot of HCl, no? Hydrochloric acid. So acidity is very, very high, sort of pH 1 to 2, no? pH sort of pH range is between 1 to 4, right? Very, very acidic, right? It's sort of in the pH 2 range. So this is the stomach enzyme pepsin. This is pH 7. The small intestine enzyme sort of work in the slightly to the basic side around pH 8, right? Around pH 8. So trypsin, chymotrypsin, enzymes are there. So this stomach acidity is because of HCl. So how do we have this small intestine එකේ basic ගතිය තියෙන්නේ කොහොමද? How does stomach inside sorry, small intestine has this sort of a basic nature? Because of, yes, bicarbonate. Bicarbonate from where? Bicarbonate from where? Pancreas. Pancreatic bicarbonate, right? Bicarbonate from pancreas. Pancreatic juice, right? Here is again, no, what's the other thing? There's no pH decline in enzymatic activity. Let's see. There's pH range. You take this pH range for him, 0 to about 4 again. That is the pH range. So what is the definition of pH range? Narrow range of pH in which a particular enzyme catalyzed reaction takes place. Right? So pepsin can't work in this pH, trypsin can't work in this pH. Simple. They have their own pH values. So what happens then? This highest pH, for example, for pepsin pH 2 and for trypsin about pH 8, those are called what? Optimum pH, the pH at which the highest rate of reaction is there. So what happens if you have the pH going above or beyond? What is going to happen? Alteration of chemical bonds involving in forming the enzyme substrate complex alter. There is going to be alteration of the bonds forming the enzyme substrate complex. That is it. So what I have to say? How can pH lead to the decline in enzymatic activity? Due to the alteration of chemical bonds involving in forming enzyme substrate complex. That's how pH can cause decline in enzymatic activity. Always it's a decline, right? When pH goes this way, it is also a decline. When pH goes this way, it is also a decline. Right? pH එක වැඩි වුණත් ප්‍රශ්නයක්, අඩු වුණත් ප්‍රශ්නයක්. Right? Okay. Right. Clear up to now? So we come to the last part of the structure. We come to the last part of the structure. I'm going to teach a bit of theory based on the structure essay, and also there's some MCQs. So the MCQ point practice today is, and also sort of the theory point that I'm going to focus on today, right? මේ දිගටම theory තමයි කරගෙන ආවේ, right? I was talking about the theory in detail, but I'm going to talk in detail about, in particular today, yes, C4 pathway. Okay, C4 pathway, let's talk in detail. Because there's a structured essay, 3 structured essay questions that we need to answer. You know, in C3 plants, there's a wasteful process called what? Photorespiration. In normal C plants, there's a wasteful process called photorespiration. Our carbon dioxide accept that is RUBP, no? Right? Carbon dioxide අල්ලන්න කෙනෙක් ඉන්නවනේ in our Kelvin cycle. Ribulose 1,5 bisphosphate. This RUBP plus carbon dioxide reaction is catalyzed by who? Rubisco. Rubisco enzyme. Rubisco, no? So if this reaction happens, this is called carboxylation reaction because carbon dioxide is being captured. If that happens, what are we going to get? We are going to get an unstable intermediate 6 carbon because meata 5 carbons, no? This has 5 carbons, this has 1 carbon. We are going to get a 6 carbon intermediate, but it's unstable. So it's going to break down into 3 phosphoglycerate, 3 PGA, 3 carbon. Make a proper reaction, the nice reaction, the good reaction. Right? But the thing is, the Rubisco is not faithful, no? He is not very faithful. So he can do another reaction that is not good. What is the other reaction he'll do? Instead of CO2, it can react RUBP with oxygen. This is going to produce what? 1 3-PGA, and there's another product called 2-phosphoglycolate, right? That is not in your syllabus. So a 2 carbon product. It's called 2-phosphoglycolate, but it's not in your syllabus anymore, right? Parna resource එකේ තිබ්බ. So this is altogether 5 carbon. Make a problem එකක් නේ, right? So we are not fixing carbon. Photorespiration is a problem. Net loss of CO2. That's why we got the C4 path to solve this problem of Rubisco being unfaithful. Rubisco මේ වැඩේ හරි නෑනේ. Yeah, he's cheating, no? He's cheating on RUBP with not the proper one, oxygen. So to do that, what did evolution come up with? A division of labor. Whatever is, we're going to separate oxygen from Rubisco. Rubisco to oxygen ළඟට එන්න දෙන්නේ. What is the example that I usually take? If you are going to study and let's say watch movies in the same room or play games in the same room, it's going to be a problem, no? Right? Because you are going to prioritize, obviously, your entertainment over studying, right? So if you study and watch movies in the same room or play games in the same room, it's a problem. The proper thing is not going to happen. So if you study in a separate room and you have your computer or the game wherever in a different room, then there's some control, no? There's, because I'm in this room, I'm going to study, in this room I'm going to play games or watch a movie. That's what we are going to do. We are going to have 2 separate cell types that are going to do the 2 parts of photosynthesis. There's going to be a central vein. I'm going to draw the leaf anatomy, and around it there's going to be a one cell type, one type of cell. These are called what? These are the cells specialized to do Kelvin cycle, and around them we have the other specialized cells, mesophils, bundle sheaths and mesophils. Okay. So bundle sheath are inside, mesophils are outside. Not clear, no? Is it laggy online, people? No, right? Mesophil cells. So what are their specialization? What they specialize to? Mesophil is specialized to do photosynthesis. Mesophil is specialized to photosynthesis. Bundle sheath is specialized to do Calvin cycle. Division of labor. වැඩ දෙක දෙන්නෙක්ට දීලා තියෙනවා. Right? So what is the product of photosynthesis? Output? Yes, we are going to get ATP aromeva. But photosynthesis produces oxygen, right? Because of the water breakdown, no? Photosynthesis produces oxygen. But because this photosynthesis happens in a different cell than the cell that the Calvin cycle is happening, RUBP or Rubisco doesn't get into contact with oxygen because it's in a different cell. You are playing games in a different room, right? That's how the C4 cycle works. And there's another method. Whatever carbon dioxide that comes into the mesophil cell, CO2 comes into the mesophil cells. No? Whatever carbon dioxide that comes into the mesophil cell, that carbon dioxide will be delivered by the mesophil into the bundle sheaths. There's a CO2 delivery system. Carbon dioxide delivery system. On තියාගන්න your carbon dioxide කියලා they are going to give it to you. Give it to the bundle sheath cell. So come to the structured essay question. What are the structural adaptations in C4 plants for photosynthesis? Structural adaptations of C4 plants. Write down the answers. Right? Write down. Okay. Structural adaptations in C4 plants. C4 plants. Write down. First time. Compare C3 plants. Bundle sheath cells of C4 plants have expanded physiologic functions. Usually, if you take a C3, these bundle sheath cells are not functioning. They are not usually functioning. They are just there to provide protection to the central vein. This is a vein, okay? Xylem and phloem, this is a vein. In C3, they don't have much of a function. So what is the first adaptation? In C4, bundle sheath cells have expanded physiological functions. They are enlarged and have a higher organelle content. Bundle sheath cells are larger, higher organelle content. They are larger and have a higher organelle content. Then mesophyll cells and bundle sheath cells are tightly interconnected by high number of plasmodesma, right? So in between these bundle sheath cells and mesophyll cells, you have plasmodesmata. plasmodesmata. Higher organelle content, larger and tightly connected by plasmodesmata. Then finally, we have to say bundle sheath cells enclose the vascular bundle and they are in themselves surrounded by mesophyll cells. This arrangement is called what? Kranz anatomy. Kranz anatomy. Okay. If you want, you can draw this. So those are the structural adaptations. Then, what are the differences between grana found in mesophyll cells and bundle sheath cells? This is another comparison that we have to do. Okay. Is it raining online, people? You have connection issues. We have to compare. I'll erase this, right? Photorespiration. So we now know we have come up with a solution that is the C4 pathway. So in the C4 pathway, if you zoom in closer, so now we only talked about the two cell types now. So if we zoom in closer to the bundle sheath and mesophills, even close, right? අපි bundle sheath එකටයි mesophyll එකටයි තව ළඟට ආවොත් we zoom in particularly into where? Into their chloroplasts. Bundle sheath versus mesophyll cells, chloroplasts. Let's zoom in. What are the differences between please turn off your cameras, okay? Okay, right. Okay. If you write Kranz anatomy only, yeah, I mean, you're going to get 1 mark. That's it. We have a lot more to write, no? So what I was saying, yeah, we have to talk in detail about the different features of what are the differences between the chloroplast, right? What are the differences between the chloroplasts, all right? So if you come to bundle sheath chloroplasts, all right, they are specialized to do what? The Calvin cycle. So if I draw you a chloroplast, inside the chloroplast, you have membrane system. Inside the chloroplast, you have membrane system. What do you call it? Membrane system? Membrane system that contain the photosystem. Contain photosynthetic pigments. Stack මේ වගේ නිකන් ගොඩ ගොඩවල් වගේ stacks. These are called what? Grana. නේද? grana no. These are stacks of thylakoid, no? Thylakoid membrane මෙම stacks වගේ නිකන් අර ගෝනි 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 උඩ ගහලා වගේ, right? have you seen නිකන් හාල් කඩේක වගේ the stacks of rice ඒ වගේ. So these grana are doing what? They are doing photosynthesis, particularly the light reaction. I'm sorry, this is not right, right? It was the light reaction of photosynthesis, no? Light. Right? So these grana and the thylakoids are doing what? The light reaction. What is doing the Calvin cycle? What Where does the Calvin cycle happen? In this stroma, no? The stroma or the jelly-like material of the chloroplast. So if you are a bundle sheath cell that is doing only Calvin cycle, do you need grana or thylakoid membrane? No, you don't need. So grana, we say few or less differentiated. Right? Online people do this bundle sheath and mesophil difference. Right? I want you to write down reviews. A4 sheets staple කරලා write down the reviews. Write down now the difference between the chloroplast of bundle sheath and if you have taken printouts, staple කරන්න. It's or if you can better buy a book separate for paper. So this is the theory part I'm doing today in detail. So bundle sheath of chloroplasts of bundle sheaths contain few or less differentiated grana, or sometimes grana are absent. Grana ඇත්තෙම නෑ වෙලාවට. There's no grana. But if you come to a mesophyll cell chloroplast, it is specialized to do what? The light reaction. So to do light reaction, you need a lot of thylakoid. The grana are large, highly differentiated. Grana are large and highly differentiated. Yes, and I forgot to mention something. Here, in the bundle sheath cells, there are few grana, or the grana may be even absent. But if they have grana, a තියෙන grana වල there's only PS1. There's only photosystem 1. There's only photosystem 1. No photosystem 2. Any photosystem 2 net that I'll do in a different day. The linear electron flow. I want you to just remember. Can you remember? It is in the photosystem 2 where water breakdown happens. Water breaks down. මෙහෙම වතුර දුන්නහම you get oxygen. Where that happens? In photosystem 2. Right? So oxygen is bad. Why? For oxygen, if you have oxygen, we are going to do photo respiration, not the proper Calvin cycle. So PS2, you can't have photosystem 2, it's very bad. Right? So that's why even if they have grana, only PS1 is present, no PS2. Okay. In the other hand, in the mesophyll cells, grana are large and highly differentiated for the light reaction. So that's what you have to write. What are the differences between grana found in mesophyll cells and bundle sheath cells? For that question, that's what you have to write. Okay. Yeah, you can. So there's a question. So you can write about chloroplast in the second question. No? Question number 4 they're asking what are the difference between grana. So you don't need to write that in the above question. What are the structural adaptations in C4 plants? You don't need to write about chloroplast because intergranal lamella these grana are connected by sort of these connections. Intergranal lamella. right? Final question. State an active that reaction that takes place in the C4 mechanism. Right. So the final part I have to mention is how from the mesophils that are outside, no mesophil තියෙන්නේ එළියනේ. So if you take, this is a leaf. මෙහෙම leaf එකක් නම් මෙහෙම දිගට තියෙන්නේ cross section එක. How this carbon dioxide that is getting in contact with the mesophil is delivered to the bundle sheath inside. There's a carbon dioxide delivery method. That's what we are going to talk. That is called the C4 pathway, right? Okay, right. So let's talk about that reaction. Clear? Make the comparison online people. What is, if you summarize, bundle sheath cells have few or less differentiated grana, or the grana are absent. In the grana that they have, only PS1 is present. There is no PS2. In mesophyll cells, grana are large and highly differentiated. Right? You have to, embed them in your memory. Right? So this CO2 delivery system, it's actually a series of reactions. It's a series of reactions. So that's what these C4s, other than RISCO in the mesophil cells, there's another carbon dioxide accepting molecule, phosphoenol pyruvate. නිකන් අර carbon dioxide පිළිගන්න receptionist ලා දෙන්නෙක් ඉන්නවද, right? Other than RUBP, there's another one called PEP, phosphoenol pyruvate. So this PEP combines with the carbon dioxide from outside to produce what? Oxaloacetate. PEP, how many carbons? PEP carbon කීයක් තියෙනවද? PEP has only 3 carbons. With this carbon dioxide, this has 4 carbons. Right? So this whole thing happens in the mesophyll cell. තාම mesophyll cell එක ඇතුළේ ඉන්නේ. Right? We are still in the mesophyll cell. The problem is this oxaloacetate is not very permeable. එයා නිකන් travel කරන්න වැඩි කැමති නැහැ. He doesn't like to travel that much. So what needs to be done? This oxaloacetate has to go into the bundle sheath cell, but it doesn't like to travel. So we have to convert it into a more permeable way. That is either malate or aspartate. Better, its malate is common. This malate will go into the bundle sheath cell below. Right? Malate will go into the bundle sheath cell below. Then what happens to this malate in the bundle sheath is it will convert back to PEP. It will convert back to phosphoenol pyruvate. Sorry, it will convert into pyruvate actually, just pyruvate. And it will release the carbon dioxide. This pyruvate will be converted into PEP, and this pyruvate will go back into the mesophyll and it will become PEP. And for this process to convert pyruvate into PEP, you need ATP. පොඩි energy එකක් දෙන්න ඕන. නිකම්ම pyruvate you can't convert to PEP. Right? Following so far. So in the mesophyll cell, there's a separate carbon dioxide acceptor, that is PEP, that will combine with carbon dioxide to form 4-carbon oxaloacetate. Oxaloacetate will convert to malate, that is more permeable. It will come into the bundle sheath cell. Malate will become pyruvate. pyruvate will be converted to phosphoenol pyruvate back again by ATP. This carbon dioxide will be what? This carbon dioxide will be accepted by Rubisco. That Rubisco, that will be, I mean, this is the Calvin cycle. Right? What are the carbon numbers? Malate 4 carbon, pyruvate 3 carbon, phosphoenol pyruvate 3 carbon. There's a nice enzyme here that is called phosphoenol pyruvate carboxylase. මෙතන රුබිස්කෝ වගේ මෙතන ඉන්නවා pep carboxylase. So these two reactions take place in the two separate cells. I'll erase this. All right. My reactions take a set in the two different separate cells, right? So the bottom cell is what? This is bundle sheath. Malate and pyruvate come back to plasmodesmata. If you like, you can quickly take this down. Pay special attention to this ATP that is converting the pyruvate to PEP. Special attention. No? So that brings us to the end of the structured essay paper. Right? End of the structured essay paper. Any questions? I talked about this. You cannot write about, you don't need to write about chloroplast to the question number 3 because why? They are asking in the question number 4. And there's enough detail to talk about Kranz anatomy itself without talking about chloroplast. Right? You can say bundle sheaths have expanded functions and they are tightly interconnected by plasmodesmata. Basophils are outside, bundle sheaths are inside. There's a lot to describe. So you don't need to talk about that when you are describing the Kranz anatomy. You can compare the chloroplast later. So if you want, you can take down C4 pathway, how carbon dioxide, the carbon dioxide that is here is delivered to here. Robiscoගේ අත්තටම ගැල්ල දෙනවා here have the carbon dioxide. Don't be distracted by oxygen. Don't be distracted by oxygen. Please don't do photorespiration. Okay. Come to the MCQ paper. Come to the MCQ paper. Okay. Come to the MCQ paper. For short answer questions, there's a theory part. I'll just give you answer. And no need to discuss around that much, right? MCQ number 1. Monomer of inulin is, yes, what is the monomer of inulin? Give me 1 minute. So first question, monomer of inulin is what? Fructose. Fructose, what are the things you know about fructose? So is it an aldose? It is a component of sucrose, right? Fructose, what are the things you know about fructose? It's a ketose. It's a ketone sugar, so it's not an aldose. Is it a component of sucrose? Yes. Glucose and fructose from sucrose? Yes. Is it a non-reducing sugar? Fructose? All monosaccharides are reducing sugars, right? Glucose, fructose, galactose, all monosaccharides are reducing sugars. Is it a pentose sugar? No, they are hexoses, 6 carbon, no? Right? It's a hexose that is insoluble in water. No. Answer number 2, no? Yes. What is the answer? Answer number 2, no? Yeah. Right. Which of the following regarding amino acids is correct? Which of the following regarding amino acids is correct? Every amino acid has an asymmetric carbon atom? No. Except what? Please write down. Please write down reviews. Who has a symmetric carbon? Glycine, right? You can find glycine as a triple carbon. Only carbons control formation of hydrogen between amino acid albumin. Albumin is protein. Yes. What is the level of ionization? Ah, yeah, right. So this comes under one of the essays. We'll keep that. Keep the second MCQ. Okay. I'll talk about orders of protein structure Taggala because there's an essay question. Okay. Come to third one. Golgi apparatus is a stack of tubular sacs. No, it's a flattened sacs. No, is it tubular? Gold produces vesicles containing hydrolytic enzymes. Does Golgi produce hydrolytic enzymes? I think yes. Golgi. Second, I'll come back, okay? Because there's an essay with that I'll discuss. Golgi produce lysosomes. So lysosomes, yeah, lysosomes can be hydrolytic. No? Yeah. Receives vesicles from rough endoplasmic reticulum by its transphase. No. Quickly, I'll explain. Right? What are the two phases of Golgi? Golgi තියෙන phases දෙක මොනවාද? What are the two phases of Golgi? Golgi has two phases. Right. One phase is facing the rough endoplasmic reticulum. This is media, right? This is the receiving phase. This is the coming out phase. So this is coming in, going out is photons. So receives reticulum by its cis phase. Huh? So correct it. Cis phase එක තමයි receiving phase. That's how you cis phase. Synthesizing, collecting, modifying, packaging, distribution of proteins and benefits. There is no synthesizing, right? Golgi does not synthesize, right? That's what makes it wrong, right? That's what makes it wrong. Golgi does not synthesize. හැබැයි අනිත් ඔක්කොම කරනවා. Collect කරනවා, modify කරනවා, package කරනවා, distribute කරනවා, ඔක්කොම කරනවා. But not synthesize. Contributes to forming the cell plate by vesicles formed by the cis phase. That's what it makes it wrong, right? It is not, the vesicles are produced by what? The transphase. So the vesicles are going out of the transphase. මෙහෙම තමයි බෝල බෝල යන්නේ එළියට transphase. බෝල බෝල ඇතුළට යනවා into the cis phase. බෝල බෝල එළියට ඇවිල්ලා these sort of vesicles are coming out of the transphase. So correct it. Contributes to forming the cell plate by vesicles produced by this transphase. So what is the best answer? Answer number 2, no? Produce vesicles containing hydrolytic enzymes. 4. Which of the following is correct regarding enzymes? Toxins are often inhibitors that bind to enzyme through weak bonds. Obviously, no? සාමාන්‍ය toxin එකක් කියන්නේ එහෙම නිකන් ලේසියෙන් it's toxic, no? So they usually bind through covalent permanent bonds. Right? Toxins, they are usually irreversible inhibitors. Okay? Many competitive inhibitors are irreversible? No. Usually competitive inhibitors are reversible. How can you reverse a competitive inhibitor? I'm erasing this. Huh? How can you usually reverse a competitive inhibitor? By adding substrate. Competitive inhibitor. Right? So competitive inhibition can be reversed. You add more proper substrates. If you add substrate, the competitive inhibitor will be reduced. Because substrate is not coming. So correct it. Many competitive inhibitors are reversible. No? Correct. Enzymes reduce the energy difference between reactants and products. So these are chemical kinetics, chemistry, right? There are reactants and there are products. These are reactants, these are products. This is the reaction. This is the energy difference. මොකක්ද ඒකට කියන්නේ delta H ද? Delta H, no? What this is the energy difference between reactants and products. There's an energy hurdle that we need to jump. That is activation energy. What does the enzyme do? Enzyme comes and reduces this activation energy. So EA අපි කියමු EA1 කියලා. But whatever the enzyme does, this energy difference is the same. ඒකේ වෙනසක් නෑ. So there's no reduction in the energy difference by enzymes. You can write down as a review there. Enzymes reduce the activation energy. Enzymes reduce the activation energy. Enzymes reduce the activation energy. Vitamin B is a component of enzyme, coenzyme NAD. Yes. Right. Vitamin B is a component. So number 5, bind substrate to a regulatory site increases the capacity by the enzyme showing cooperativity. Cooperativity means the definition of cooperativity is what happens if substrate binds. And yes, the catalytic activity increases. So the substrate is binding. So where is the substrate usually bind? The substrate binds on the active site. Substrate are related to the enzyme, right? So if you have a certain enzyme, What is the example we take for cooperative? What is the example that we take for cooperative? Hemoglobin. Hemoglobin. Hemoglobin is composed of 4 subunits like this. Hemoglobin is composed of 4 pieces. We call them subunits, right? Hemoglobin is composed of 4 subunits. Because why? A single hemoglobin can carry 4 oxygens. A single hemoglobin can carry 4 oxygens. So the thing is, what happens when one oxygen binds to one of these active sites? The affinity of the other active sites for oxygen increases. Again, other active sites start to like oxygen more because one oxygen was bonded. So this is going to send signals. We have a signals to the other active site, other subunits saying to increase their affinity. So in the other active sites, the affinity increases. So binding of substrate molecule to where? A substrate always binds to the active site. So correct it. Substrate to the active site increases the catalytic activity of the enzyme showing cooperate. Right? So the answer is 4, no? 5. Some events related to metabolic processes are given below. Which of the above events can be seen in the C4 pathway? What can you see in the C4 pathway? Strictly in the C4 pathway, oxaloacetate to citric acid කියන එක මොකක්ද තියෙන්නේ? That is in the citric acid cycle, no? B, malate to pyruvate. That is yes. B is good. Decarboxylation. You can say malate to pyruvate is a decarboxylation. Hydrolysis of ATP, yes. Where? Aparadi, I just erased. By pyruvate to pyruvate to phosphoenol pyruvate conversion, I told you. Remember, Kila, right? There you have ATP hydrolysis. So D is okay. Regeneration of RUBP by G3P. I mean, that is not strictly C3, C4 path, that is part of Kelvin cycle. No? So BCD, answer number 2. BCD, answer number 2. Which of the following regarding cellular respiration is correct? Question number 6. Which of the following regarding cellular respiration is correct? Okay. Pyruvate molecules produced during glycolysis enter the mitochondria passively. No. Okay. I'll talk about cellular respiration as a theory topic. I mean, as a separate topic on a different day. Today was what? C4, right? So pyruvate enter the mitochondria actively, by active transport. So correct it by active transport. In the citric acid cycle, 4 CO2 molecules are released for each glucose. What do you think? That is correct. 4 CO2 per glucose. Per glucose, yes. Because there's 2 places where CO2 is released. Among the chuttakar, remind kanna gonetta. It's not mage hitarat thai, right? What do you feel in the citric cycle? Acetyl CoA is fed. Who is the acceptor? Oxaloacetate here. You produce citric acid. This citric acid will be converted back to oxaloacetate. What are the products released? For one acetyl CoA, there's going to be 2 carbon dioxides released. Because why? Citric acid is 6 carbon, oxaloacetate is 4 carbon. කොහෙදි හරි carbons දෙකක් has gone missing, right? So 2 CO2s produced, 3 NADH, 3 NADH, 1 FADH2 and 1 ATP per 1 acetyl CoA. If you take 1 glucose, that is 2 acetyl coenzymes A, that means these products will be doubled. Statement number 3, 2 ATP molecules are used to transport NADH molecules from mitochondrial matrix to cristae. No, there's no such thing. Respiratory quotient of protein is 0.7. No. I'll talk about RP also, RQ. RQ means what? Yes. CO2. CO2 so CO2 evolved divided by oxygen. No, oxygen divided by CO2. No? Amount of oxygen, is it the other way around? Yeah, sorry. CO2, volume of CO2 divided by volume of oxygen. Volume of oxygen absorbed versus, so volume of CO2 evolved divided by volume of oxygen absorbed per respiratory substrate, right? So if you want to respire, let's say a mole of glucose, carbohydrate, you need carbon dioxide release and CO2 oxygen absorb. You will it is fat. You need oxygen. You need oxygen metabolize or break down fat. So for fat, it's what? What is the respiratory quotient for fat? That is 0.7. That is 0.7. Protein medeke mother, that is 0.8. Remember like this, for fat, you need a lot of oxygen compared to the carbon dioxide to metabolize your fats. Glucose is equal, C6. If you write down the balance reaction for glucose, මේ පැත්තෙන් 6 carbon dioxide දුන්නාම අර පැත්තෙන් මේ පැත්තෙන් 6 oxygens දුන්නාම you get 6 carbon dioxide from the other side. So කැපිලා යනවානේ. They get cancelled. So the ratio is 1. Glycerol enters the citric acid cycle as acetyl CoA. Yeah, glycerol. What happens to the glycerol part of fatty acids? No, it enters glycolysis. No, there's a chart at the very end of your metabolism. Write this chart, go through it. What happens to the glycerol part of fat? It enters glycolysis. Fatty acids enter as acetyl coenzyme. So you can write down there, small review. Glycerol enters glycolysis as GP. Glycerol enters glycolysis as G3. Glycerol enters glycolysis as G3. Next slide. which is regarding origin of life on Earth and evolution of biology. Biological diversity. Um, Earth's neutral atmosphere favor the synthesis of organic molecules. What do you say? Neutral atmosphere. No, right? Earth's, as I remember, Earth's reducing atmosphere. No, Earth එකට තිබ්බ reducing atmosphere එකක්, as I remember. Yes, correct it. Earth එකේ atmosphere, it was a neutral. It was reduced. Inside the protocell, very sorry, it's the protocell, not photocell. Nuclear materials are surrounded by a membrane. In the protocell, was there anything surrounding? No. Protocell එක කියලා කියන්නේ it's a very simple cell. Even now the modern prokaryotic cells don't have a membrane surrounding the nucleic material. So protocells did not have. With the origin of the first photosynthetic organisms, the amount of atmospheric oxygen began to increase. Yes, photosynthesis produces oxygen. Simple, obvious. Anyone bio කරන්න නැති කෙනෙක්ට. After the first eukaryote appears, a great range of unicellular forms. No, right? After the first eukaryotes, what did you get? You start to get multicellular forms because eukaryotes are more advanced. Right? Then ethani unicellular story is now ending because now what we have eukaryotes. Ancestors of arthropods began to colonize land after the colonization of fungi. Who first colonized land? First colonization of land by animals, fungi, plants, and animals. Yes, plants and fungi colonize the land by interacting with each other. No, so afterwards we got the arthropods. So answer number 5. No, answer number 5. Ancestors of arthropods began to colonize the land after colonization by fungi. If you want this exact answer, third lesson, page number 74. That paragraph. What about 3? Oh, hmm. Take both. Take answer number 5 and 3 both. Better answer is answer number 3. No. Yeah. Take both 3 and 5. Select the response with the correct match regarding history of classification. Right. These are direct theory points. Right. Direct theory points. Nothing much to discuss. Who introduced the 5 kingdoms? Even I can't remember. Who introduced the 5 kingdoms? 5 kingdom, H. Whittaker, no? Robert H. Whittaker introduced the 5 kingdom. So then classification according to hierarchy of species, genus, order, and class. That was Linnaeus, no? Carolus Linnaeus. 3 domain classification, Carl Woos. Introduced the taxon phylum. Who introduced the taxon phylum? That is again Carolus Linnaeus, right? Who introduced tax and phylum? Ah, no, sorry, Ernest Haeckel. Very sorry. Ernest Haeckel introduced tax and phylum. Sorry, introduced two kingdoms of plants as plants and animals. Two kingdoms. That is again Linnaeus. So, what is the best combination? So, with wobbly. What is the best combination? Carl Wooster, we can give A to S. AS, there's two answers. Then Ernest Haeckel introduced the kingdom phylum. I'm sorry, taxon phylum. Ernest Haeckel, taxon phylum, BT. Again, same. Carolus Linnaeus. CQ and CU both correct. Then Whitaker, five kingdom classification. D. Hmm, BR. What do you mean? Ernest Haeckel is introduced kingdom protista. Ah, yes, 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 yes. He introduced kingdom protista. No, yes. Answer number 5 is best. Right? Can you see? So I suggest you make a short note. I can give you one for the history of classification. Make a short note. In the revision class, we are going to do that. Okay, we are going to make a short note for the history of classification. It won't get confusing, right? History of classification. We are going to make a short note in the revision class. Okay. Next one. Several types of wood and their features. Let's keep it. I want to summarize these. I want to teach the theory around this next class. Okay. Wood and so question number 9 and 10. Please keep. Okay. We are going to discuss them with the proper theory surrounding it. I need some more time than today. All right. I'll make a note of it and I'll discuss together with secondary growth. Okay. Secondary growth theory part when I'm discussing, I'll do it. Right. 11. In many plants, sugar from mesophyll cells are transported to phloem. How? ටිකක් නිකන් just apply some logic. මුකුත්ම දැනගන්න ඕනේ නැහැ. So, in the mesophyll cells, we make the sugar. In the phloem, we transport the sugar. So, if you are thinking about the amount of free sugar, where do you have more free sugar? At the transporter, no? Because in the phloem is modified to transport the sugar. This is the phloem tube now, right? And there's a mesophyll cell very close by. The mesophyll cell is making sugar, yes, but the phloem is the one transporting. So, if you take the sugar concentration, it is high in this mesophyll cell. I'm sorry, in the phloem. The sugar concentration is high in the phloem. So, can you transport this sugar from mesophyll to the phloem in a way that is passive? No. You have to load, we call it the phloem loading. We have to load this phloem actively. Nimita, I like to take this example. And so before that, so phloem eකට මේ පැත්තෙන් we load the phloem and on the other side we unload the phloem. No? So this is the starting point. This is the end point. We'll talk about this phloem translocation in a different way, but I like to use this example. example like you see, so even if you are doing some construction at your house, you know, like construction, you order some sand, very good again, you order some sand from the hardware for doing this. So the sand lorry comes, and what does it do? It just tips all the sand into at your home. No? Tipper wrecking home unload. So unloading is easy. But how do you think order to the sand with by hand? No, things all they have loaded by hand, individual showers. So that's why I like to tell you the sugar loading, the flour loading is a very active process. But it's very easy to unload. නිකන් අර ටිපර් ලොරි එකෙන් have you seen unloading ටිපර් ලොරි එකේ තට්ටුව මුස්සලා ඔක්කොම වැලි ටික ලස්සනට හලාගෙන යන්න පුළුවන් just using the lorry. කවුරුත් අතින් බාන්න ඕනේ. No one needs to do it manually, right? So just keep that example. Phloem loading is active. Phloem unloading is usually a passive process. So sugar from mesophyll cells are transported to phloem by active transport. Answer number 5. Which of the following occur during ethyl alcohol fermentation? So the numbers are a bit wrong, right? This is an ABCD type question. Okay, I hope you have noticed. This is an ABCD type question. There can be more than one correct answer. Ethyl alcohol fermentation, because we need to know this in the, I'll draw the, let's say, the flow chart of ethyl alcohol. What happens in ethyl alcohol fermentation? What happens in ethyl alcohol fermentation? That's how we make alcohol, no? Yeast use kalla industrially. How we make alcohol. So everything starts with what? As usual, glucose in, no? That is our substrate. The starting point is glucose. Glucose will be converted to what? Pyruvate. Pyruvate. This process is what? Glycolysis. Glycolysis. Then what happens to this pyruvate? It is converted to a mid product called acetaldehyde. Acetaldehyde. Acetaldehyde will convert. Right? So this process, acetaldehyde to ethanol, conversion is done by who? NADH. That is produced by the glycolysis itself. So there is NADH here. It will convert back to NAD plus, and it will convert acetaldehyde to ethanol. So one glucose will produce pyruvate. Pyruvate will be converted to acetaldehyde. Acetaldehyde will be reduced to ethanol using NADH. This process then let's focus on the carbon numbers. Carbon numbers, glucose has 6 carbon, pyruvate 2 pyruvate, no? So 3 carbon. Acetaldehyde has only 2 carbon. So what happened to the other? CO2. You have heard of when we are making alcohol, there's a lot of CO2, right? Our fermentation process එක beer එහෙම හදද්දි there's a lot of bubbles. And also we use this CO2 production in where else? Bread, when we are making bread. Right? පාන් හදද්දි අර palm පිපෙන්න what we use to raise the flour. පිපෙන්න දාන්න baking powder. Baking powder contains yeast, right? When yeast is converting the glucoses present in our bread dough into ethanol, small amount of ethanol is produced, it is making carbon dioxide. So when you heat this carbon dioxide, it is going to expand. That expansion is going to make air holes. So when you are baking the bread, these air holes are going to get bigger, 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 and the bread is going to come texture. Like only like this, right? It's going to be like this, but bread is very crumbly, right? It's full of air because of this carbon dioxide released, right? Ethanol. So what is the question? NADH produced during glycolysis is used? Yes, A is true. One molecule of glucose is converted to one molecule of ethanol. No, you are going to get two ethanols. Right? One glucose is going to make two ethanols, two acetaldehydes, two carbon dioxides also. Pyruvate is converted to acetaldehyde? Yes. Molecules of CO2 are released from one pyruvate? No, pyruvate only one CO2. Quickly take this down. Ethyl alcohol fermentation reaction. Quickly take this down. Final MCQ. Some features of protists and the organism that process those features are given below. Right, a bit of protists. Presence of a pellicle. So we have Euglena, amoeba, and paramecium in under protozoa. Algae එකක් තියෙනවා, that is Gelidium. So pellicle is present in Euglena and paramecium, right? If I can remember, Euglena and paramecium, right? Pellicle එකක් තියෙනවා. Hold on. Euglena has a pellicle. Paramecium has a pellicle, yes. Then contrary only amoeba, I think, and also paramecium. All three have Euglena, paramecium, and amoeba has all three has contrary. Phototrophs. But Euglena can be photoautotrophic as well, but we consider it as a mixotroph. So not the best answer. Gelidium is the best. Locomotion by flagella. Yes, Euglena has flagella. Euglena has flagella. What's the answer? DP CS. There are several good answers, no? So let's find out the answer. So pellicle A, you can give Euglena and paramecium. Paramecium is what? R. What is the second feature? Contractile vacuoles. For this, you can give P, R, and amoeba also. P ඔය තුනම දෙන්න. C වලට you can give photoautotrophic වලට you can give gelidium and euglena දාලා බලමුකෝ නේද? Euglena is P. D, locomotion by flagella, there is only euglena, no? So P. So what are the answers having these combinations? A and B, no? A and B, A, B, D has D and P as the answer. DP තියෙනවා A and B වල. A, R is okay. B, Q is okay. CS is okay. So answer number A is fine. Check B. AR is okay. BR is okay. CS is okay. DP is okay. B is okay, no? C you can't take. D, AR is okay. BQ is okay. CP is also okay. ABD, no? A, B, and D statements are correct. A, B, and D statements are correct. Right. Let's discuss the structural levels of electrons only. You want me? Final essay. I'll send the vascular tissues of plants. That's a very straightforward essay. I'll send the marking scheme for both actually. I'll be sending the marking schemes for both. So this protein essay, I'll just quickly go through the theory part. Levels of protein organization, quickly come to the resource. How many protein organizations levels do we have? I know these are very basic. That's why I didn't start with these in the revision class. Right? Revision class, okay, I didn't start with these because honestly, you are tired of this. But anyway, they are going to be questions. We'll discuss them a bit while later, bit closer to the exam, when you have forgotten them. How many structures do you have? Right? Right. It's a sequence of linear unique sequence of linear range and minus. It's like a chain of what it wants. Right? These are peptides. It's a unique sequence. If you run the amino acids, connected by peptides, primary structure. Secondary structure can be either what? It can be like a sheet, right? The beta pleated sheet, right? It can be like this, which is the beta pleated sheet. So it can be a helix, the alpha helix. two forms, right? There's a beta ben that is not in your syllabus, right? So how is this secondary structure formed? Intramolecular hydrogen bonds within the oxygen of the carboxyl and the hydrogen of the amino. Very important to remember, within the molecule, intramolecular H bonds. So if there is a oxygen here of the CO group and there's a hydrogen here of the amino group, there's going to be a hydrogen bond. Tertiary structure, the secondary structure will fold and coil. Right? It's a ball of, it can contain මෙහෙම helix තියෙන්නත් පුළුවන්. It can contain beta pleated sheet areas as well. It can again contain helixes. It can contain beta pleated sheets. Like that, it's a globular protein. Right? Precise, compact, unique, functional, and 3D shape. Right? It occurs between what? Interactions between R groups. Interactions between R groups. Right? Finally, when this is formed, you get more than one polymerase. Right? So more than, usually more than two. One O, right? One O. So global proteins, they were like this. They are connected by inter and intra both. They are connected by inter and intra bonds to form a polymerase. Okay. So those are the levels. I will send you the essay marking schemes and the vascular tissue essay question as well. That is a very straightforward question. So online people, this is the first paper discussion. We are only discussing sort of half papers and also from very early parts, because I'm sort of we want you to rewind these sections at the earliest as well. But at the same time, we are sort of kickstarting the paper classes, right? We are going to switch on to full papers. When we are doing that, the discussion will be longer and all of your papers will be marked and given, right? Even online papers. But if you can come to the institute, ISM Nugegoda, and you can write the paper within the paper slot, writing slot, then you can hand over and you can get them marked, right? The advantage of that is you have the writing interval, so you can focus, right? So I advise, if you can come, but if you can't with the distance, then obviously you can send us the PDF. We'll mark and give. So that's the paper class, how it's going to go. We have another whole set of programs planned for the revision class, right? I'm going to start something that sort of a YouTube live series that I'm going to cover small topics in the syllabus that you can watch whenever you are bored, right? Kammalikamak kawam balanna puluwan විදිහේ I'm going to start a small YouTube series that is part of the revision class. So we have a lot of programs planned in the upcoming 7.5 months almost. So yeah, we can finish for the day. Any questions? Any questions you want to ask? Then we can finish. Thank you.